Okay, so I'm going to talk about some practical considerations for data analysis. I'm going to start with some, just so we can all be on the same page, start with some basic scattering theory and diffraction geometry, and then talk about how crystal properties, the quality of your crystal, the order of your crystal, how that all affects the data analysis procedure, how your data is actually affected by that. Okay, so to start with, Ed touched on this a little earlier, so I won't go too much into it. For X-ray diffraction, we have X-rays coming in with a given wavelength, generally used monochromatic wavelength, and they interact with the electrons around the atom, and they re-radiate in the spherical wave. And as you have multiple atoms, they interact with each other. And so what happens is you have these diffraction patterns that show up on your detector. So for example, just a simplified version with two atoms, you end up with either constructive or destructive interference, depending on whether or not the waves are in phase. And so at certain angles, you end up with constructive interference, where the waves superpose. And in other angles, you end up with destructive interference. So you end up with no signal. And so we use this information to essentially back calculate to what the orientation and position of the initial atoms are, where they are positioned relative to each other, so that we can determine the structure of molecules. Now, for crystallography, or macromolecular crystallography for proteins in particular, we can represent protein structures in different ways. So this is a three-dimensional model of a protein structure using what's called a ribbon diagram. So we have alpha helices, beta strands, and loops. But this obviously isn't how proteins actually look. This is just a geometric construct to help us visualize things. In reality, they have a surface defined by the van der Waals radii, where each atom has its own radii and its own electron configuration. And so what we do is when we actually look at the structure of proteins, what we're actually looking at for X-ray diffraction is the structure of the electron density function of that protein. Now, what happens is that with an electron density function in 3 dimensions, you can perform a Fourier transform of it, and you end up with what's called the molecular transform. So on the right here, we have the equivalent of a single molecule diffraction pattern, which is essentially a projection of that molecular transform onto a 2D detector. And so you notice that this does not look like conventional crystallographic diffraction patterns, and that's because this is a continuous distribution from one single molecule. What happens with crystals is that that underlying molecular transform gets sampled by the lattice packing of the lattice arrangement of the molecules. And so what you're actually looking at when you see those diffraction patterns is the underlying molecular transform sampled at those Bragg locations. And so we use that intensity information to then back calculate to our electron density function for a molecule. So here's just a quick example of how this actually works. So we start in the upper left with a single molecule, a very simple molecule, and you end up with this continuous distribution. And if you add another molecule, you end up with an interaction because of those waves being added together. In certain areas, you get destructive interference. In certain areas, you get constructive interference. So we end up with a modulated diffraction pattern. If you have more molecules, say in 2 dimensions, you end up with, instead of these sort of slices through the diffraction pattern, you end up with slices in both directions, in both axes through the diffraction pattern. And you can see as we go on and we have a full crystal, you end up with just diffraction peaks. So imagine expanding this to 3 dimensions. That continuous molecular transform that you start with in 3 dimensions actually ends up being sort of compressed into these discrete reciprocal lattice points. And so we use this information with X-ray diffraction through some basic geometry. So we have an incident beam coming in from the left here with a given wavelength. And when you have a crystal, you end up with a series of lattice planes. Joe talked about that earlier. And so these lattice planes actually end up acting, you can model it as though it were an actual reflection off of those lattice planes. They scatter in a particular direction, like that. so that you end up with scattering in a particular direction depending on the configuration of the lattice planes and how they're orientated relative to the incident beam. And so you can think of it as actually having your incident wave vector, your scattered wave vector, that's Ki and Ks, and you end up with a diffracted wave that's perpendicular to the orientation of those lattice planes. And so we call that wave vector K. So with a crystal, we end up having a series of different lattices that are, or different lattice planes, a set of lattice planes that are made up. So we have a lattice, and you can actually see you can create a series of different lattice planes because of the orientation of those molecules. And so each one of those set of lattice planes will give off a diffracted wave in a different direction. Now, we use what's called the reciprocal lattice, or it's essentially the Fourier transform of the real space lattice, because it makes a lot of the math simple, and it makes the theory a little bit simpler. So the spots in a detector correspond to those reciprocal lattice points, and what you do is you end up measuring the intensity of those reciprocal lattice points in your 2D detector and build it up into 3 dimensions by having either a rotation series or serial crystallography, and we'll get to that in a little bit. So every point on a reciprocal lattice represents the set of planes in real space. So every time you actually see a point, a spot on a detector, that represents the intensity corresponding to one set of reciprocal lattice planes. So it is a reciprocal lattice, right? So one of the things that you can think of is that larger unit cells end up with smaller spot separation. So this is a little animation. We have the plane wavefront coming in from the left here, interacts with a line of atoms, and they scatter, and they form these constructive interference lines. If you have a 2-dimensional set of atoms on a plane, you end up with your reciprocal lattice shown on your detector. Because it's reciprocal space, if you have larger unit cells and you expand your unit cells, you end up with spots that are closer together. And if you make them smaller in one dimension, it makes them closer in one, or further away in one dimension of the whatever dimension that's getting smaller. If you add in other atoms, it actually keeps the same pattern except for the intensities. It modulates the intensities of that pattern, because what determines the location of these spots is that crystallographic packing, whereas the intensities of those spots are determined by the 3-dimensional arrangement of the atoms within the unit cell. Okay, so everybody's heard of Bragg's law. It's essentially the equation that makes our field possible. And it's essentially an equation that tells you what angles satisfy the most constructive interference. So if you set up a basic geometry here where we have an incident plane wave coming from out on the left here, it interacts with a series of atoms or molecules on a lattice, and you end up with a phase difference, a path difference between the scattered wave of one atom on a plane compared to the scattered wave of the neighboring atom. And so what happens is if this theta, this d is just right, you'll end up with a scattered wave that adds up in phase in a certain direction, only at a particular angle. However, if it's out of phase, if your angle is not correctly corresponding to d, given by this Bragg equation, n lambda equals 2d sine theta, then you'll end up with a scatter wave that's out of phase, and so you won't see it, essentially, because there's just not as much intensity. So what happens is that 2 theta angle is where you end up with your constructive interference. High-resolution diffraction patterns end up resulting from a series of planes that have small d spacing. Again, it's reciprocal space. So if you have a very small d spacing, then you end up with a very high angle. Okay. So the reason why I went over all this relatively basic stuff is so that we could talk about the Ewald sphere. So the Ewald sphere construction is a geometric construct that allows you to visualize the relationship between your lattice planes and the incident wave vectors and your angle of diffraction. And so essentially what it is if you think of what we mentioned earlier of our incident plane wave, we just rearrange the vectors, you end up with k as ki minus ks. So what happens is if you actually map out all the different possible diffracted KS's for different orientations, you end up with a circle, at least in 2 dimensions. You end up with a circle that actually maps out all of the angles that will show you diffraction. It's essentially the sphere of Bragg's law. So what happens is that when you have a reciprocal lattice, which might actually be on the next slide, you have a reciprocal lattice, instead of having a continuous distribution where somewhere there's some electron that's on the Ewald sphere, in this case, because of our reciprocal lattice being concentrated at certain points, we only get scattering at certain directions. Only when we, this is a rotation series on the right here, so for conventional, say, synchrotron crystallography, or you have a crystal sitting on a goniometer and you rotate it through 3-dimensional space. And what that allows you to do is actually get the reciprocal lattice points to pass through the Ewald sphere at different orientations so that you can sample all of 3-dimensional space. Okay, so once you end up with your series of diffraction images, you have to be able to describe this pattern. So we need a mathematical way of looking at this pattern, identifying what spots are what the intensities are. And the first step to that is indexing. Well, I guess actually the first step is finding what the peaks are in the first place. But once you have your peaks, you can index. Now, crystals themselves, or protein crystals at least, for chiral molecules, there's only 65 different ways of arranging chiral molecules in a crystal lattice in 3-dimensional space. And so because of that, we have a limited number of space groups, and so this restricts the possible arrangements of spots on the diffraction pattern. And so you can actually use this information to help your indexing. You know that there's only so many different possible arrangements, but there's obviously essentially a continuous distribution of unit cells. So a larger unit cell with the same space group will end up spreading your spots apart. A larger unit cell will end up making your spots closer together. A smaller unit cell will end up making your spots further apart. And so you use indexing, where programs essentially look at the location of all your spots, and it attempts to determine which set of unit cell parameters, what space group, what symmetry, and what your crystal orientation is for that particular pattern. And so it does this so that it can assign each value or each 2-dimensional position on your diffraction pattern, which corresponds to a 3-dimensional position as you do your rotation series or your full set of data. It assigns a value of HKL, which is essentially the reciprocal lattice Miller indices, to correspond to those in 3-dimensional space. And so that's what our indexing does. It essentially determines what those spots are and where they're located in reciprocal space. So the next step is integration. So integration, once you know what those reciprocal lattice points are, you can integrate over the spot. Essentially, you have a spot that looks like this thing. And you end up measuring what the signal is, what the number of photons in each one of those pixels. And you integrate over the total area of the spot, and you determine what some background is around it. And this tells you your signal-to-noise ratio for that particular spot. So you need to know these intensities because, as I mentioned, this all works back to that initial molecular transform that you're going after, right? So we're looking at a reciprocal lattice, sort of that discretized view of that molecular transform, but these intensities are still fundamentally based on those structure factors from your molecular transform. So the final result from your integration step is that it yields a 3-dimensional array of HKL with corresponding intensity, and generally a signal and a noise. And you use that because that correlates to the magnitude of the structure factors for structure determination. Now, I'm not going to get into it for the purposes of data analysis, but that's only part of the way there. Because we have only the intensities, we actually lose what's called phase information. And so you have to take the second step of solving the phase problem, which is obviously not a trivial problem. Okay. So all of that was to help you understand why we use the Ewald sphere to help us visualize the relationship between reciprocal space and essentially Bragg's law. So what ends up happening is that for different spots, you have, for different crystallographic forms, you have spot shapes. Okay, so the spot shape is determined by a series of different parameters, and I'll get into those in a little bit. But certain spots are not necessarily just a sphere. You know, if you have certain crystalline disorder of different types, you can actually smear out your spheres and end up with, you know, ellipsoids or some kind of shape that's modulated by what's called a shape transform. You end up with a shape that does not necessarily give you all the data if you cut through a different part of the Ewald sphere. So this is an extreme example here, but essentially the two things that I want you to notice with this slider is that the crystal properties determine the three-dimensional profile over the reciprocal lattice point, whereas the beam properties and the beam geometry are what determine the Ewald sphere. And so if you have normally we assume that we have monochromatic light, but it's not perfectly monochromatic. There's actually a distribution, or with XFELs, there's actually a very noisy distribution. So you end up with some kind of distribution of wavelengths. There's angular divergence. There's different factors that are incorporated into the Ewald sphere. And so what happens is you end up having multiple Ewald spheres for every single one of those different, essentially beamline geometries, different wavelengths or whatever. You end up with, say, the lower end of your distribution of wavelength. You end up with a larger sphere. If you have a higher end of your wavelength spectrum, you end up with a smaller sphere. And so you end up with, this is the origin of reciprocal space. I should have labeled that. But if you have this sort of shell-like construct, you end up sampling reciprocal space in different ways depending on where you are in reciprocal space. So, for example, if you're further out where we have this extremely exaggerated shell here, you end up collecting the full intensity, the full possible diffracted intensity from that shell, from that reciprocal lattice point, because it's fully within the Ewald sphere. However, over here, you end up with only a small sliver that's actually within the confines of the Bragg law, within the shell of the Ewald sphere, and so you only end up with a partial reflection. This is important because, particularly for XFELs, pretty much every reflection is a partial reflection because the waves, the beam is very well collimated and extremely monochromatic, so you end up with essentially partials for every reflection. So that's a big problem because ultimately what we want to know is what the full reflection value is, what the full intensity value is. Okay, so now that we've gone over some of the basic theory of how Ewald sphere works, I'm going to talk about how crystalline order can affect those different parameters within Ewald sphere. So the purity of your crystal, the disorder of your crystal, one of the most fundamental parameters it's based on is the purity of the crystal growth solution. So it's one of the most significant factors that affect the crystal and the diffraction quality. And if you're unable to reproduce your experiments, oftentimes it's because of impurities in your crystal growth solution. It can affect the rate at which your crystal grows. It can reduce or increase the solubility. You might end up crashing out sometimes and having clear solutions other times, or wherever in the phase space. You can either suppress or enhance nucleation because different solutions are going to be in different locations in your nucleation, crystallization diagram. It can change the morphology of your crystal. Typically, with general lab procedures, we end up with somewhere in the neighborhood of 5 to 10% impurity concentrations. And for very high quality commercial purities, we end up with somewhere around 1%. Okay, so what types of impurities are we talking about? So there's a wide variety of different macromolecular impurities. One of them is structurally related genetic variants. So if you have different splice variants or isoforms, or mutations in a expressed protein, then these are all going to be small little defects from molecule to molecule to molecule that are going to spread across your crystal. And so these cause these are a form of impurity. They're causing a less order across the complete crystal. Another form is post-translational modification. So you have glycosylation, deglycosylation, whatever kinds of post-translational modifications you can think of. You can also have partial denaturation. Certain forms of proteins tend to not like other solution conditions, so it might denature. Through the process of crystallization, you can actually get fragmentation and things like that. You can also have oligomerization, so there might be different oligomeric states of your protein depending on crystal solution conditions, concentration, say, and all those end up being as different forms of that fundamental unit of your protein molecule that you want to be identical from cell to cell to cell, from unit cell to unit cell. You can also get other structurally unrelated molecules. You might accidentally pull down some other proteins that stuck to your column for whatever reason. And so all of these different macromolecular impurities cause some kind of disorder in your crystal. Okay, so a lot of times these impurities cause defects. They can be defects such as dislocations or cracks. You saw some pictures of that earlier. Twinning is obviously a big problem sometimes. You can have polycrystalline or amorphous aggregates that end up making data collection more difficult. You can have increased mosaicity, and we'll talk a little bit more about that soon. And you can also end up with decreases in resolution, among obviously other things. Okay. So now we're going to get into some of the effects on the data of these different impurities and these different types of disorder. So there are ways that you can actually characterize the quality of the crystal straight from the diffraction data. One of the most prominent ways of doing this is just resolution. If you have poor resolution, that's some kind of indication that there's disorder in your crystal, right? Higher resolution data means you have order to very, very small d spaces. Another mechanism of identifying this is what's called the B factor, or the temperature factor. It's also called the atomic displacement factor. And essentially what that is a way of describing the motion of atoms within a crystal relative to some equilibrium position. And we'll talk about the different types of atomic displacement you can have. And so just a quick example of that over on the right here. If you have essentially a B factor of 0, and say you take a spherical atom, you end up with this distribution that has a higher intensity, particularly at high angles, than if you have a B factor, a temperature factor. You're essentially taking that atom and smearing it over some region of space, and so it acts as though you have a larger volume with the same number of electrons, so you get less scattering. And finally, the major way people do this is through mosaicity. And we'll talk about that more. Okay, so the first one was resolution. So in general, intensity falls off very rapidly with angle for macromolecular crystals. And so what we call resolution is generally defined as the maximum scattering angle at which diffraction peaks can be reliably measured. Either you measure this through R merge or CC star or signal to noise, something like that. But it's the highest resolution that you can get, the highest 2 theta, the highest angle that you can get reliable measurements of intensity. Reflections, what they actually tell you, as I mentioned before, are the magnitude of the Fourier coefficients of the electron density. And so what that means is that if you have a series of different waves that are corresponding to different d spacings, this tells you, the highest resolution tells you what the highest frequency Fourier component is in the crystal. And so that tells you what your highest d spacing is, what the correlated signal that you can actually get, the constructive interference, the highest reliable signal that you have corresponds to the shortest d spacing. And so that's the highest frequency Fourier component, otherwise the shortest wavelength Fourier component. So if you think about it in terms of real space, resolution is the ability to distinguish between features in electron density. So if you have, some kind of, paired Gaussian peak, some kind of bimodal distribution, say you're looking at the location of 2 atoms, if your resolution is poor, then you won't be able to distinguish the position of those 2 atoms. And if it's very high, then you'll be able to see those 2 peaks very clearly. Low resolution means that there's very few short wavelength Fourier components that have been measured reliably. which then results in a decreased ability to distinguish those features because you're just not sampling it finely enough to be able to tell the difference between the atomic locations. Okay, so what are some of the factors affecting resolution? This is a very, very brief list, but there's a lot of different factors that affect resolution, but the primary ones are the overall intensity scale of the diffraction pattern. And usually this is indicated by the intensity of low resolution reflections, because at low resolution most crystals are at least ordered enough, and B factors tend to not contribute as much. The fall off of the intensity is a function of the scattering angle. So as I mentioned, because the things like B factor will actually cause your intensity to decrease, it decreases differently at different scales, at different resolutions. So as you can actually measure that fall off as a function of the angle and get some kind of idea of what's going on. Resolution is also obviously affected by background scatter. So we're looking at signal to noise. So you have your signal, which is defined by your crystal properties in the order of your crystal and the intensity of the beam, essentially, among other things. But you also have background, either from diffuse scattering within the crystal, or you can have instrumental noise. And so all these different characteristics of your experimental setup can introduce more background. And so our goal is to have a high signal-to-noise ratio that we can. Okay. So let's talk quickly about the crystalline characteristics that affect resolution. Hold on one second. Okay, I'll try and speed through the rest of this. Okay, so crystal volume affects your resolution because a larger illuminated volume means that there's more electrons that are scattering, so there's more unit cells, and therefore the intensity of the spots is higher. The unit cell size will also can also affect resolution. If there's a larger unit cell, there's generally fewer unit cells for the same crystal illuminated volume, and therefore you'd end up with lower intensity spots. Solvent content is a big one. If you can see on the right here, this is what's called the Matthews coefficient. So if you have a smaller solvent content, you can actually see if you bin this according to resolution, that you end up with a statistically significant dependence on resolution and solvent content. And obviously the character and extent of internal disorder, which is what we're going to get into next. Okay. So I mentioned B factor, and B factor is a measure of the fall-off rate of intensity as a function of resolution. In this particular case, we're measuring what's called the Wilson B factor. So essentially what you do is you take the average intensity of several different resolution bins for all your different reflections, and you plot them as a function of log of that intensity versus essentially angle squared. And so what you end up with is what should be a relatively flat line. There's a lot of assumptions that go into this, but from the slope of this line, you can determine your B factor. And so this essentially tells you the average B factor, the average atomic displacement across the atoms in your crystal. Now, there's a lot of assumptions that go into this that make it a little bit inaccurate, but it's a great way of assessing initially the quality of your diffraction data, and at least your B factor. So B factor not only has a measurement from the data itself, but you can actually model it. And so every atom in your resulting PEB file has a B factor associated with it, some kind of atomic displacement value that tells you how much the atom is moving, or maybe not even moving, but it could be some kind of static disorder, differences from unit cell to unit cell. Now, B factor is generally insensitive to crystal defects such as dislocations and cracks, and the reason for this is because those are types of long-range order, and B factor is generally affected by short-range order, on the order of like maybe a few to several unit cells. And because of that, it's very highly sensitive to different variations such as conformational variability, chemical modifications, displacements of or rotations of atoms from molecule to molecule. Typical structures have atoms that range in B factor anywhere from maybe 5 to 100 angstroms squared from protein crystals, whereas like small molecule crystals, you're looking at less than an angstrom squared kind of. And this ends up with a displacement factor of anywhere from 1/10 to 1 angstrom, just to give you an idea. Okay, and so the final thing was mosaicity. So mosaicity is essentially a way of describing how a crystal is actually subdivided into a domain of different blocks, what we call mosaic domains. So these mosaic blocks are essentially, because of impurities and imperfections in the crystal, you end up with a block of very similarly orientated crystals here. This block is rotated a little bit differently. This block is rotated a little bit differently. And so we can actually model that by a series of essentially small domains. And you end up that each domain, because it's a slightly different angle, ends up having a slightly different angle for where Bragg's law is satisfied. And so it has a slightly different reflection angle. And so if you think about that on the Ewald sphere, what essentially that means is you're smearing out your spot over you're smearing out your reciprocal lattice point, which results in the diffraction pattern spot being smeared out as well. And so this is a few examples of that. So we have on the top here very low mosaicity, less than 1/10 of a degree. And as you increase in mosaicity, you can see that you gradually increase the spread of those spots to the point that at very high mosaicity, it actually becomes difficult to separate the spots, and so you get unreliable intensity values because you don't know which photons are from which spot. This oftentimes occurs during cryocooling. So one of the major things that if you're doing low temperature like cryocooled crystallography, you want to make sure that your cryoprotectant is not interacting somehow with your protein to cause it to be poor quality in the cryocooling process itself, because you're essentially shocking your protein with different range of temperatures over time. You flash freeze it to minimize this, but it still can cause some issues. Small crystals often exhibit less mosaicity, so when we do nanocrystallography, because a lot of times this crystal is smaller than the actual size of the mosaic domain, you end up with less mosaicity. Okay, so this is just a quick recap of those things visually. So we have different types of disorder in a crystal. So over here we have positional or thermal disorder, where atoms might be in slightly different locations. You might have different loops. Orientational disorder, where the molecules or the unit cells are, the molecules within the unit cell might be in slightly different orientations. Maybe those crystal contacts aren't as firm in certain unit cells. You can have conformational disorder, where maybe you have a loop in one conformation in this position in this unit cell, and a loop in another conformation in another unit cell. And those three are all short-range types of disorder, because locally you can see that there's differences from unit cell to unit cell to unit cell. Whereas a type of long-range disorder that generally affects mosaicity are things like grain boundaries or dislocations and cracks, because you can see that if you kind of zoom into the short-range order, this unit cell is very similar to this unit cell, even though this block is different than this block is different than that block. So those are the types of things that distinguish short-range disorder from long-range disorder. Okay, so I'm just going to talk real quick at the end here about serial crystallography. This is BioXFEL. So while all of these parameters essentially that I've discussed are true for both conventional, rotation series crystallography and serial crystallography, there's certain unique things about serial crystallography that can be either helpful or problematic. So serial crystallography is when you have a solution of small micro or nano crystals that are injected into an X-ray beam. And because they're in a solution, they're in different orientations. Whereas on a synchrotron, if you've got some kind of goniometer, you're rotating it through, so you know the angular spread, you know the angular rotation of each one of those shots. Because these are random orientations, we don't initially know what the orientation is when we see the diffraction pattern. And so when it comes to indexing, that's actually a big issue with indexing. You need to know your orientation very, very well to make sure that you're estimating those intensities and your partiality correctly. So as I mentioned, for XFELs, essentially all the reflections are partial, and this is due to the very monochromatic beam because it's a laser. And so this leads to very thin Ewald sphere shells, and so you end up with most reflections just being slices. And so you have to collect tens or even hundreds of thousands of images to be able to build up those full intensities. And the way we do that is what's called a Monte Carlo method. Essentially, you just have so many copies of that reflection that you can build up a distribution of those reflections and make an estimate of what the full intensity actually is. but it requires a lot of patterns. And so this makes the method less, less exciting, I guess, or less, amenable to many proteins, because a lot of proteins can't express that high. You can't get MIGs or mils of crystalline solution sometimes. And so this makes the whole indexing process more difficult. However, new attempts or methods to analyze this data allow you to actually model the various aspects of the crystal, beam size, beam parameters, in order to actually try and throw all of those into equation and accurately assess what the full intensities are, and get accurate orientation so that you can end up with just a few thousand patterns rather than tens of thousands of patterns. And real quickly, because you need a new crystal for each image, this means that the crystal to crystal to crystal parameters are slightly different. So you might have different mosaicity from one crystal to another. You might have different short-range order. You might have the volume of the crystal is different. The cell parameters of the crystal can vary, and so you end up with these distributions. And so this is one of the things that the Monte Carlo method actually kind of takes care of, is that you've got so many patterns that essentially it averages out all these differences. So one of the ways that you can actually help this and not require as many patterns is by controlling the sample heterogeneity. So things like filtering your solution of crystals, or doing some microfluidic sorting, things like that, which is what this paper is being submitted on. You can see that if you actually control, if you have a broad distribution of crystals, where you have a lot of different crystal volumes, you end up with a much quicker fall off of the quality, whereas if you have a very narrow distribution of crystal sizes, you end up with much higher quality data out to higher resolutions. Okay, just some quick bullet point summaries. The key thing here is that the optimization of the crystallization conditions, including, very importantly, the purity of the sample, are paramount to the high-quality diffraction patterns, high-resolution diffraction patterns that we want. So if your diffraction pattern isn't high resolution, try other crystallization conditions. That's probably one of the easiest things to do. Other things that you can do to increase the quality are things like dehydration or seeding. They can help improve the mosaicity. Crystal packing, this is one of the more theoretical points, but I think it's very important. The crystal packing arrangement determines the location of the diffraction spots, and the atomic positions determine the intensity of the diffraction spots, because you're sampling that underlying molecular transform. And understanding these different reciprocal lattice relationships and essentially the Ewald sphere construction I was talking about, how unit cell parameters and symmetry affect these things, are very much required to help optimize your data collection strategy. Rather than go on, I'll stop there. And if anybody has any questions, I can answer them quickly. We're a little behind.